All right, so again, welcome. I'm Dr. Kevin Basile. I'm the Director of Didactic Education at Arcadia University in our Physician Assistant Program. Arcadia University is again located in Glenside, Pennsylvania. We actually have two separate campuses. Our main campus, where the university itself is housed, is in Glenside, Pennsylvania, which is sort of just northeast of Philadelphia. We also have a secondary campus, or an expansion of our program, which is located in Christiana, Delaware, which is about 20 minutes or so just south of Wilmington, Delaware, which is the capital of the state. Today for the webinar, I'm going to go through basically 5 different steps. First, just quickly talk about our physician assistant program so you have an idea of how we initially presented gross anatomy to our physician assistant students. Talk about, again, how we presented lectures, our lab sessions, how we assessed our students' knowledge, and then really the next steps, the evolution. As Jake introduced me, we've been using Anatomage now for about 4 years, and we've sort of been evolving how we've incorporated it into our program. And I just want to provide those different steps that we did in getting it integrated into our program, and then using feedback from our students, feedback just in terms of our own faculty experience. We continue to revamp it, and then with every new version of Anatomage that comes out, trying to incorporate some of the newer features that they provide for us on the Anatomage tables. I'll put a little disclaimer in there. When I was first asked to do this presentation, I just made sure that Anatomage knew that, you know, with the good and the bad, I was going to present my experience with the Anatomage table. And so if you do have any questions or concerns, I'm more than happy to answer them for you in terms of just our own personal experience there. We started with, again, our program as I described. We have 2 campuses. Again, they are separated probably by about 50 or 60 miles. And within the 2 programs, if we were to combine them together, we have between 100 and 106 graduate students. About 56 of them reside at our Glenside, Pennsylvania campus, and about 50 of them reside at our Christiana, Delaware campus. Our physician assistant program lasts 24 months. The students have an entire year devoted to didactic curriculum. And that starts really right around Memorial Day, and usually ends around mid-May the following year. And then they start their clinical rotations, again, right around mid-May. And then they'll do that for a year, and they'll graduate really right around Memorial Day of the following year. So the way we initially were providing our students with gross anatomy in terms of lecture material, we'd have our students meet for lectures 3 days per week, and they would have about 3 hours worth of lecture per day. So roughly speaking, it was about 9 hours per week. The lectures were provided at an off-campus location that was sort of equidistant between the 2 campuses. But even though the campuses themselves were about 60 miles apart, where we were off-site, which is where we were housing our human cadavers, with the regional traffic patterns and everything else, it was still taking anywhere between 45 minutes to an hour for our students at each campus to actually get to the off-site location. And so, you know, again, similar to, you know, probably those California attendees at this meeting, you can imagine just how bad our traffic is around here on the East Coast in the Philadelphia area. So that was our first challenge, is what we were going to do with our lectures. The second challenge was exactly what were we going to do with our lab sessions and dissections from the perspective of what we were doing in our program 5 years ago. And so again, because our students were coming to that off-site area for lectures, and again, that's where our human cadavers were stored, the students then would also have their dissections 3 days per week, and their day was allotted to about 5 hours worth of dissection. So again, Monday, Wednesday, and Friday, our students were off-site for about 8 hours, really just doing gross anatomy. And then the other 2 days per week is where we would place all of the rest of our didactic curriculum. And then from a third challenge perspective, we would provide our students with 4 written exams and 4 practical exams. Each exam, the written exam and the practical exams, were each devoted an hour for them to each be accomplished. So again, we were looking at 2 hours of examination processes that were occurring. Now, our gross anatomy program within that didactic year was provided over a 12-week time frame. So that was sort of the constraint that we had in terms of approaching or utilizing the Anatomage table into our program. So to summarize, as we were looking into incorporating the Anatomage program instead of utilizing cadavers, we had to sort of address all of these different circumstances into how we would then incorporate it into our program. And one of the challenges, obviously, is when those lectures were being provided offsite versus onsite, how would that impact the rest of our didactic lectures? Because one of the initial challenges we had was when our leadership was looking at, why should we move over to a digital platform to provide our students with dissection? One of the issues was obviously the storage of cadavers, the ability to obtain cadavers, and again, the time constraints that our students were experiencing in going to our anatomy sort of off-campus location. So to bring that all sort of into a campus environment and integrate it in with our didactic lecture material, I was actually tasked with converting our live lectures, which I said was about 9 hours per week, into recorded lectures. Because in moving to the Anatomage table, we came up with our first roadblock, which was we couldn't have 10 Anatomage tables like we had 10 cadavers. We needed to figure out a way for our students to be able to operate or understand dissection and again, these relationships of structures to other structures by utilizing fewer, in essence, fewer cadavers at any one time. The other challenge that we were faced with was we were having both of our campuses go to one central location for their lectures and cadaver dissections. But now what we were going to do is we brought this more into each individual campus. We would then have to provide the lectures and dissections at each respective campus, ideally synchronously. But when we initially started this process, the leadership had informed me that they wanted to actually do it sort of asynchronously, meaning that I would be present at each campus during the laboratory sessions. So in order to do that, we had to negotiate where the rest of our didactic lecture curriculum would reside. And one of the solutions, at least, that was proposed was moving from doing live lectures in front of students to actually having them do, again, asynchronous learning through recorded lectures. And then we would use the laboratory sessions as a way for the students to do question and answer of the recorded lectures, as well as then proceed forward with the dissections themselves. So I was tasked with, again, converting from a live anatomy didactic lecture format to a recorded format, and then shifting our lab dissection from 3 times a week to only once per week at each respective campus. And that even our lab dissection times, instead of them being 5 hours per day, 3 times per week, we were actually going to shorten it all the way down to 3 hours per week at each respective campus. Initially, we weren't going to change the examinations or assessments at all. We were going to keep them as 4 written and 4 practical and keep them as 1 hour each. So those were really the challenges I was faced in when I was tasked with, what were we going to do and how would we incorporate the Anatomage tables into it? So how we started to break this down was that first we sort of kept the consistency of how we would lecture to our students. We broke our regional anatomy down into 10 different areas. We started with the thoracic cavity. And unlike a traditional, I guess, anatomy dissection, where typically you start students on the back of the torso and then proceed on into, the thoracic wall or perhaps down into the extremities, we were trying to synchronize our anatomy dissection with the didactic material, the other didactic material that our students were learning. And so in our pathology course, which our students also have during the summer semester, we actually start talking about first the integumentary system, but then we proceed into the thoracic cavity, and we start talking about the structures and components within the thoracic cavity from a pathophysiologic perspective. So we wanted to review the anatomy with our students really prior to them getting exposure of discussion within our pathophysiology course. So using the integumentary system part of our pathology course as sort of the buffer, we were able to present or sort of keep our anatomy 1 week in advance of what the students would then review in their pathophysiology course. So within the 10 regional anatomy structures or regions that we would talk about, we started with the thoracic cavity. We would then proceed down into the anterior and posterior abdominal wall. And then we'd move into the actual visceral organs and neurovascular components of the abdominal cavity. We then would proceed up into the central nervous system, where we would talk about the brain and the spinal cord. We'd then review the head and neck, followed by the sensory organs, at which point we would then move to the posterior aspect of the torso and review the back. And then we do the upper extremity. We'd move into the pelvis and then the lower extremity. And we designed it that way, as you'll see in a few more slides here, based on then how we started putting things together from an examination standpoint. As we talked about these different regional parts of the body, we then also started doing pre-recorded lectures, really, again, just using the PowerPoint slides that we had utilized during live lectures, except we were using a digital platform to record the lectures, and then we were posting them within our digital, Blackboard classroom system. So that was sort of how the first year evolved, which was in 2018, and we modified it a little bit in 2019, but then obviously the COVID pandemic hit, and we had to pivot a little bit more. And at the time, sort of between 2019 and 2020, we were already in the process of, rather than our students simply listening to a recorded lecture with PowerPoint slides, we actually started recording, sort of displaying the Anatomage table onto video screens that were in our lecture hall, where we then were able to have our university's camera system and recording system actually capture the table as it was being displayed on the screens at the front of the lecture hall. And so we were utilizing really both a PowerPoint presentation slide along with the Anatomage table recordings, really right around when the COVID pandemic hit. And it was sort of during that time also that Anatomage actually, their technology and their computer programmers and everybody started developing what was known as their Table on Cloud system. And so we were able to also utilize that to help do some of our recorded lectures. As I mentioned, we were shifting to, again, the use of Anatomage tables. And so our university purchased basically 4 Anatomage tables. 2 of them were going to reside at each of the campuses. And we provided our students with 24/7 access to the tables so that they could asynchronously learn and have some guided direction via myself and some of the other faculty that were involved. I think for us, one of the more challenging spots was, again, how do you get 100 to 106 students through a regional anatomy dissection in a 3-hour block of time. And the way that we designed how we would do that is, roughly speaking, we have between 50 or 56 students at each campus. And so we broke up or assigned those 50 to 56 students into 3 separate groups, and we're just going to call them group A, B, and C. And you can imagine if we were doing a 3-hour block with each group, that would be a 9-hour day. So very similar to how we were doing the live lectures and dissection. We were still devoting basically an entire day per campus to run students through their sort of dissection aspect of the course. And in order to provide some equity within our groups, we didn't always have the A group coming in first thing in the morning and the C group always being the last group in the afternoon. We actually would rotate our A, B, and C groups every week, so that, week 1, it might be A, B, C, but week 2, it was groups B first, then C, then A, and we'd sort of cycle that through over the course of the 12 weeks. So every group was either an early morning group or a late afternoon group at least 4 times in our 12-week session. As I mentioned, we did 3-hour lab sessions per group. And then the other unique aspect of what we then introduced was what we would call the reverse classroom lab facilitation. And so here what we would do is we would actually assign students in the group every week to actually be a lab facilitator. And we gave them, again, access to the Anatomage table to sort of build their lab facilitation, their identification of structures, the discussion of these structures. They had access also to traditional models consisting of the bone boxes, anatomical joints, and plasticized organs. And within each of these A, B, and C groups, we actually would divide those groups one more time. So we'd subdivide them. Each group roughly had about 16 to 18 students. So when we subdivided it, we basically had 2 groups of students within the A group. So we'd have like group A1 and group A2. They each had about 8 students each. And then 1 student from each of those 8 students was the ones that were assigned to be a lab facilitator. So in any one week, we had anywhere between 12 and 16 students that were actually assigned to be a lab facilitator for any one particular week. And with the Anatomage table, what we were able to do is, as you're doing a dissection, you can actually save your dissection into what is called a preset. And what actually allowed, that ability allowed for collaboration, not only within one campus, but actually across campuses, because our students could download, quote unquote, a dissection preset and send it to a student electronically at the other campus, any of the other 12 to 16 students, and they could build upon it and then save that as again, another preset, and then they could send it back to, again, another student. And so collaboratively, they work together across both campuses. In order to help guide our students a little bit more, what we would do is we actually created what I would call a lab worksheet. Within this lab worksheet, this is, again, just an example here of the upper extremity, where, again, we asked our students to identify not only the location or the name of a muscle, but again, its origin, its insertion, its muscle action, the nerve innervation, and the blood supply. And so we would give them a list of every structure that we wanted them to include in their 3-hour sort of presentation to the other students in their group. Again, they had access to our pre-recorded lectures. They had their own textbooks, and then they also had the different functions within the Anatomage table, which again, over the course of the last 4 years has improved in terms of additional features that have been incorporated onto the table that actually allows the students to really almost have self-directed learning, where a lot of this information, as soon as one of these structures is identified, Anatomage allows then sort of for a table to appear on the table that actually provides a lot of the functionality, the innervation, and the blood supply to a particular structure as well. So it allows the students really to do some self-learning. We just sort of directed them in terms of what specific structures we wanted them to review. And so again, you can see, again, this is just a list of, again, what was typically on their lab worksheet. And our students knew from day one what week they were going to be a lab facilitator, and they knew what region of the body they would be responsible for. So from day one, they could start the process of figuring out how they wanted to present their lecture to their fellow classmates. Again, just a further continuation of the list. So you can see it was pretty extensive in terms of what we were asking the students to do. And part of this process was then also figuring out, again, looking at the research that's out there and the effectiveness of reverse classroom learning. We sort of have a side study going on trying to see the results of that reverse classroom on student performance. That being, if you were a lab facilitator for particular regions of the body, and then we did an examination, did those students who actually facilitate the lab dissection do better or do significantly better than their counterparts? So that study is actually under investigation right now, and we're starting to analyze the data. But I'll talk a little bit more about the performance of our students over the course of this transition and over the course of the last 8 to 10 years. As I mentioned earlier, we initially had 4 written exams and 4 practical exams. And what we ultimately did as we were sort of changing and modifying our program, instead of having a 50 or 55 question written exam followed by a 50 or 55 question practical exam, the pandemic sort of influenced this as well, to the fact that we actually ended up sort of merging both our written and practical exams together. And we increased it from 4 exams to 5 exams over the 12-week span. And so now our current evolution is that our examinations, our assessments are about 70 to 72 questions in length. We allow our students about 90 minutes to complete the exam, and the exam is broken down into, again, about 50 or 55 questions that are written. They include multiple choice, matching, fill in the blank. And then from a practical perspective, we use about 20 or 25 pictures pulled right off of the Anatomage table. I'll show you some examples of that. We've used pictures of the bone boxes, or sometimes instead of using a picture of a bone from the bone box, we would use pictures of bones from the Anatomage table. Same could be said for the plastic models. And then we also included some human cadaver high-res dissection pictures from really 2 different medical school programs that one was University of Michigan and one was Texas A&M University. And again, I'll show you sort of how that looked on our examinations. This would be sort of, again, some of the ways we use the Anatomage table. You can see that we would pin various structures and take a screenshot, and then we would ask our students various questions about the structures that were pinned, much like you would do in a traditional human cadaver specimen that was on an examination table. And these were some of the styles of written questions that we would have, including, again, a fill in the blank or multiple choice or some sort of matching question. So this is, again, how we built it. What you're seeing right now is actually, the review session where we identify the correct answer for our students. But that would be the style of questions that we would ask. We'd ask anywhere between, again, using Bloom's taxonomy, the lowest end of the pyramid where we were asking for simple recall, but then we'd also move up in Bloom's taxonomy to having first order, secondary, or tertiary order style of questions. So again, an example from the Anatomage table. Here would be, again, a second order question where we've identified a muscle, but yet we're asking the students to name the nerve that innervates that muscle. So they have to go through that two-step process. And this would be embedded right into our examination platform. Here is an example from the University of Michigan. They have a website that's used, they call it the Blue Link Images. It's their sort of cadaver dissection. And again, you can see here that really we weren't trying to hide anything from our students. They knew that we were using the University of Michigan website. These structures are annotated on the website. They also have an area on their website where these structures are unannotated. And what we basically did was similar to what you saw on the Anatomage table. We built pins into the picture, and then we would ask again first, second, or third order style questions based off of whatever structure we identified within the pins. And then, like I said, our course sort of progressed over time. And I want to show you just an example. Like I said, we initially used PowerPoint presentations and then recorded the PowerPoint presentation. And some of the feedback we were getting from students at the time was it was hard to see what we were talking about, even though we were using pictures from anatomy textbooks and we were looking at them from different viewpoints. There wasn't much animation going on, like real-time animation. Yes, the various PowerPoint images might fade in or fade out or things like that in order to get a different viewpoint. But again, we were looking at sort of two-dimensional images as opposed to a three-dimensional sort of product. And so one of the things we were able to do as we were recording the Anatomage table being displayed on our screens, and or when we actually were able to use the Table on Cloud system, was we were actually able to record the dissection of the cadaver on the Anatomage table. So I'm going to stop my sharing here for a second so I can switch screens. And in controlling the placement of the hand in space. In our discussion on the arm, the antebrachium is organized in a similar fashion, such that the muscles of the antebrachium that have a similar purpose and innervation are going to be grouped within the same fascial compartments of the forearm or the antebrachium. The distal forearm and wrist have minimal bulk in order to maximize their functionality, and the movements that occur down here in the hand and in the wrist are going to be controlled by extrinsic muscles that are going to be located more proximally in the forearm, which is very distant from the site of action. You can see there are very long, slender tendons that are going to extend distally into the hand and fingers in order to provide movement.
I'll move this up a little bit in the video so you can see,
the brachial vein,
again, just more animation that we were using.
We mentioned the tendon of the biceps brachii muscle inserting distally through this triangle. You can see the median nerve continuing through the cubital fossa, but we also have the radial nerve, which will start to divide into superficial and deep branches in the terminal portion of the antebrachium or forearm. Supply only extensor muscles.
So again, I'm just going to skip around here a little bit.
Which in this position,
some of the things that we would do.
If we were to remove all of these muscles and look at the deep layer, we would have 3 layers of muscles comprising the deep layer. They will include your flexor digitorum profundus, the flexor pollicis longus, and the pronator quadratus muscle. So that is an example, again, of how we started to integrate the Anatomage table into a more interactive recorded lecture for our students. So that was initially how we really started developing the incorporation of our recorded lectures into our didactic curriculum, again, allowing students to have asynchronous learning. Again, in that video, hopefully you at least got an appreciation for, again, just some initial features of the Anatomage table, where as we were going through the various muscles of the antebrachium, we were able to color them. Students were able to see the outline or the highlight of various structures. And they were able to sort of follow along with an actual PowerPoint presentation that was provided to them while they could, quote unquote, watch the video of the dissection itself, and myself as an instructor and lecturer sort of talking through the various structures with them as it went. When you first start using the Anatomage table, it seems overwhelming at first. And what we have discovered over time, and it takes a little bit of encouragement as a lab facilitator, that you could have a group of students around the table, or even within our own faculty, those that use the table often versus those that don't. And what you find is that individuals who use the table very often become what I'll call a super user. They're very fluid. They know what the different tools are, the different features of the table, and they tend to do everything. And other students or other faculty tend to sort of sit back and watch more so than anything else. And so one of the ways we got around this was, again, by doing the reverse classroom, we knew that that would encourage every student to use the table more. At the same time, it allowed us an opportunity to, I'll be the first to admit it, I'm a little bit more old school. And so by doing reverse classroom, what we've actually come to the realization of is the students, as they become sort of the lab facilitators, they've been sort of born and raised on technology. So what they were able to do actually provided even more feedback to us as faculty as other ways to use the Anatomage table that perhaps we didn't even think of. So every year, we basically have had 100 new users sort of showing us different features or different techniques that they used in their presentations. And then we would introduce those ideas to subsequent year students so that they were then more aware of other uses of the table that perhaps even as a faculty member who used the table to even do their presentations or their recorded lectures, things that I didn't know. So we found that to be a very effective way to work on this. And I see just from the survey here that many of you are on the higher end of your comfort level with technology. So I think those are some of the things that need to be considered as you're looking at how do you incorporate the Anatomage table into your program itself. The lab sessions, as I said, we did the reverse classroom part of it. I forgot to mention that during these live sort of dissections with the table, myself and one other faculty member were present within each of the different groups that was presenting at the time. And we were listening to what the lab facilitators were saying. We'd correct them if they said something incorrectly, and or if we felt that they were leaving a pertinent part of anatomy out in terms of that particular region or that discussion, we would interject and make sure that our students had that information. And then at the same time for our program, again, one of the complexities of human anatomy is, again, this volume of information without really knowing the context of why particularly a student might need to know anything about the particular structure. So the faculty were there to provide really some clinical context of why these structures are important or why the nerve supply or blood supply were important, so that, again, they would build upon it as they continued on in their didactic year and then obviously into their clinical year. As I mentioned, in terms of our examinations, they've evolved over time. Initially, again, we did the traditional written format and then the practical format where, again, our students used to walk around the Anatomage table, and from their various perspectives around the table, they'd have a particular pin that there was a small index card on the table that they would ask, that would ask them the questions. And then around the actual lecture hall is where we sort of had some of the plastic models and things like that that were sort of set up for them. When we sort of combine the written and the practical exams together, it's now all digitized. And so again, you now see that we're down to 5 examinations, 90 minutes each. And again, it's a combination of both written assessment as well as visual or practical examination formatting. One of the things that we were able to do, because this was one of the shortcomings that I felt was still existent in the Anatomage table, even though, again, they are true cadavers that were scanned and the slices were digitized together, and you have 4 cadavers built into the Anatomage table. One of the things that I was always concerned about as we were introducing Anatomage into our program was really 2 factors. One, I wanted our students to still respect human life and the sacrifices that people make in the donation of their bodies to medical education or science education. And so I still wanted our students to have that experience. The other experience I wanted them to have is still the ability to even though they've been preserved and the tissue itself doesn't feel the same as live tissue, I still wanted them to have an opportunity to feel as well as to, for lack of a better word, poke and prod human cadavers. And then the last aspect of it, when it relates to the Anatomage table itself, I didn't feel that the Anatomage table provides a viewer or a user with depth perception very well. And so I went back to our university and asked for us to at least procure a few human cadavers that we could dissect and sort of hold on to as prosected specimens, that in our 12-week session with our students, we would actually have 4 days built in where our students could come in as part of a review session in preparation for one of their 5 exams. And we would actually review on the prosected cadavers the various structures in the various regions that we had already reviewed on an Anatomage table during those reverse lab dissection days. And so that was sort of the evolution of lessons 4 and 5, where our students had the opportunity, again, to be exposed to human cadavers. They were already prosected, so they weren't necessarily utilizing their time trying to dissect and have clean specimens. And then they also had this immediate sort of review session with faculty present as they were reviewing the prosected cadavers, in order for them to identify perhaps, gaps of knowledge that they may still have had, that they could then go back and study before they actually would take our examinations themselves.